Eukaryotic gene expression. We looked last time in the last video at this level. So if chromatin is really densely packed, then the enzymes can't get at it and transcription is low. And if the chromatin is spread out like it is here, if it's not densely packed, then the machinery to transcribe it can get to it and you'll have more transcription. So there are lots of other levels at which we can control how much of a protein is actually made. So remember the whole central dogma: DNA makes RNA and RNA makes proteins, right? So you can regulate how much protein is made based on whether your DNA is available or not. Once you have available DNA, once the DNA is loosely coiled, the next step in regulating how much protein is made is this level right here, the transcription level. So if the DNA is available and RNA polymerase can get to it and can bind to it with a pretty high affinity, meaning a pretty good ability to hang on, then you're going to have lots of transcription. So we're going to look at this level in this video and actually the RNA processing level, how long RNA stays around or whether it gets degraded, whether RNA can attach to ribosomes or not. And then we'll look a little bit at whether the protein is made active or inactive to start with and how it can get activated. So we're looking at DNA, and there are parts of DNA called control elements. A control element is a segment of DNA that's non-coding, meaning it doesn't serve, in this case, as a piece of DNA that codes for RNA, which would code for proteins. So this is a piece of non-coding DNA, and it serves as a binding site for transcription factors. Transcription factors are proteins that are needed to help RNA polymerase bind. So this would be the coding piece of DNA. And this is going to code for a piece of RNA that codes for a protein. This part is the promoter. That's where RNA polymerase binds. These are called control elements. And the control elements can be proximal. Proximal means close to the gene. You can think of the word approximate. That means like, yeah, pretty close. So proximal means the control elements that are close. And these are the distal control elements. So distal, think distal, distant. These are the ones that are distant from the gene. So this little line means it can be actually pretty far away. So based on where these control elements are, we can call them either distal or proximal. And they're going to help control whether this gene gets transcribed at a high rate or not. So control elements are pieces of DNA. The control elements can also be defined based on what they do. So the control elements can be called enhancers or silencers. That's based more on function than on location. So an enhancer is involved in turning on a gene, and a silencer is involved in turning off a gene. So this would be a case of an enhancer right here, and that can be pretty far from the gene. So the gene is going to be right here. This structure right here is RNA polymerase. So remember, that's the enzyme ACE that you need in order to transcribe the gene. RNA polymerase binds onto this piece of DNA, which is called the promoter. And then there are proteins called general transcription factors that I'll define for you in just a minute, but those help RNA polymerase to bind onto the promoter. These are called mediator proteins here. This is called an activator. And instead of a general transcription factor like these, it's a specific transcription factor, and it binds to an enhancer. So this would be an enhancer. And these proteins would all be working together in order to help this gene get transcribed at a high rate. So transcription factors, these are proteins that bind to the control elements. So proteins that turn specific genes either on or off by binding to a control element. Again, a control element is the piece of DNA. It can be proximal, can be distal, can be a silencer or an enhancer. So transcription factors can say go or stop. They can be activators. This B should be over here. So activators are the proteins that boost a gene's transcription or increase, and repressors are the proteins that decrease a gene's transcription. If you look at these transcription factors here, you'll notice that they have different domains. A domain is a piece of the protein, usually comes from different segments of different exons in the RNA. But anyway, this part can bind to another protein, and this part, so cool, can bind to DNA. Okay, so how do transcription factors actually work? To initiate transcription, initiate means start, eukaryotic RNA polymerase requires the assistance of proteins called transcription factors. You always need general transcription factors for transcription of any gene. A lot of times you also need specific transcription factors. So these general transcription factors are proteins, and you need them for transcribing any protein coding gene, meaning any gene that codes for messenger RNA that will then code for a protein. So how does it actually work? This is the gene. Here's the promoter. Remember, in eukaryotes, there's a TATA box, a bunch of T, A, T, A, T, 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 or whatever. So this is the promoter that RNA polymerase binds to. Here's RNA polymerase, and you can see it binding to the promoter down here. So in order for RNA polymerase to bind, it needs general transcription factors, which are these. So you can see them bonded here. It also needs these mediator proteins if you're going to have any, if you're going to have a high level of transcription. And then it also needs these. So this is part of DNA. These are pretty far away from the gene, so we're going to call them distal control elements. These are going to say go, so we're going to call them enhancers. So the enhancers are pieces of DNA, they're control elements, and they bind to activators. Activators are proteins. These are specific transcription factors that will bind to DNA. They have a segment that binds on here to the DNA and a segment that holds on to proteins, or different domains that will hold on to the proteins. So these proteins are all required together in order to do transcription at a high rate. So we've got the control elements, or enhancers, also called distal control elements. They have bonded here to the activators. The activators have bonded to these mediator proteins and the transcription factors. And the transcription factors are needed in general, and all of them together help RNA polymerase bind onto the promoter to do transcription. So we'll say they'll increase the affinity of RNA polymerase for the promoter. So affinity just means how well does the RNA polymerase bind onto the promoter. Proximal control elements, again, are located close to the gene. And distal control elements, usually these are enhancers, can be farther away from the gene. So an activator is a protein that binds to an enhancer and stimulates transcription. So here's the activator, and and it would be attached here to the enhancer. And in this case, it's pretty distant. In this case, it's actually a little bit closer, right next to the RNA polymerase. So sometimes the enhancers are not distal. All right, so putting the whole thing together, the first thing that happens is that you need these activators, which are proteins, to bind to the enhancers, which can be pretty far away from the gene. And then you need a DNA bending protein. So that does exactly like what it sounds like it does. It attaches to the DNA and helps it bind, so that helps it bend, so that these activators can come close to the mediator proteins. And then you'll also need the general transcription factors here, and they will all help RNA polymerase to bind onto the promoter to help increase transcription of this gene. So the activators have two domains. Remember, domains are the portions of DNA that come from different exons. So they have a domain here that binds to the enhancer, and they have a domain here that will bind to the proteins. Bound activators facilitate a sequence of protein-protein interactions that result in transcription of a particular gene. So these activators will actually be very specific for these enhancers, and the enhancers are very specific just for this particular gene. Some transcription factors function as repressors. So in this case, here's a repressor. It's a protein that can bind to the DNA, and it only binds to this section of DNA. And that section of DNA is called a silencer. And here's the repressor. So if this is the promoter and RNA polymerase, this one, tries to come over and bind here, it actually doesn't have, it can't get on because the repressor's here. So it's the repressor is blocking RNA polymerase. So a silencer is a DNA sequence that can bind to a repressor. So here's the silencer, and the repressor again is the protein that stops transcription. Some activators and repressors also act indirectly by influencing chromatin. So this part's kind of cool. So we just talked about if chromatin is loose, then you can regulate transcription with activators and repressors. But those activators and repressors can also come back here and influence chromatin structure. So you know how with bar bodies, for example, they're usually really tightly bound heterochromatin? But we know that a little bit of transcription does take place in the bar bodies. So maybe there are some activators that can come back to certain particular genes and make the chromatin loose enough for just a couple of genes to get transcribed. So kind of cool. Okay, this actually, speaking of cool, is the I think the coolest example of gene regulation ever. I think this will really help drive home your understanding of this. So we're looking at a liver cell nucleus. So inside the nucleus, here's the DNA. And then we're also going to look at a lens cell nucleus. So what is a lens cell? So inside an eye, here's an eyeball. If I can make it a circle. Yay. Here's the cornea, which is the clear part, and then you have the iris, which is the colored part of your eye, and the pupil here in the center. So light comes in here, and it's focused by a lens. I drew that too far back a little, but anyway, there's the ciliary muscles, I think, and they're attached to the choroid coat here, and then you have a retina right on top of that. Anyway, light comes in, and it will focus on the retina in the back. The lens of the eye is this. And the lens is clear, and the lens produces a protein called crystalline. But the lens doesn't produce albumin. Albumin is a protein. You need it in your blood. It's made by the liver, and you don't need albumin in your eyeballs, or especially in the lens of the eye. So I want to look at the liver, which is this organ that we talked about it before. It does a lot of stuff, but one of the things that it'll do is detoxify the blood. Another thing that it does is makes bile. Another thing that it does that I think we talked about it is it changes glucose to glycogen if you have high glucose levels. So anyway, the liver does a lot of stuff that's very different from your eye. And so you would think you would need different genes transcribed in the liver cells compared to the lens of the eye. So every cell in your body has the same DNA except the gametes, right? But you don't use all the same DNA in every place. So let's take a look at the activators, the proteins that you find in the nucleus of your liver cells. These are the activators, and they can attach to these particular enhancers and make this gene get transcribed at a high level. But if you look over here at the eyeball cells, the lens of the eye, the activators here are different. There's one that's the same, but the others are different. So there are a different combination of proteins, a different combination of activators. And so the enhancers here for the albumin gene don't have activators attached to them. So this gene's not transcribed in the lens of the eye, but it is transcribed in the liver cell over here. So cool. The crystalline gene, though, this has different enhancers. And these two don't have activators on them. So the crystalline gene is not expressed in the liver cell, which is good because you don't need it there. And the enhancers, though, for this gene do have activators that are present in the lens of the eye. And so that does increase transcription of crystalline, and you do get a high rate of crystalline gene expressed in the lens of the eye. Really beautiful example of how differential gene expression occurs. It's because you have these different proteins that are available to turn on some enhancers and to turn on some genes and to not turn on other genes. So you might ask next, how come these proteins are here in this cell, but different proteins are here in this other cell? And so that's a tougher question, and we're going to look at it a little bit when we look at embryonic development toward the end of this unit, and I can help give you the idea of how you sort of get that way, but I don't really have the specifics for the eyeball and the liver. Okay, coordinatedly controlled genes in eukaryotes. If you remember back to prokaryotes, Coordinated control, we looked at operons. Do you remember operons? So operons are the way that prokaryotes transcribe genes together when they need, like, let's just do an example. So we talked about the trip operon, and so there's enzyme A that's coded for this part of DNA, and enzyme B maybe has these two genes that need to be transcribed, and enzyme 3 has another gene. So these would be the genes that code for the mRNA that code for the protein, and all of those 1, 2, 3, 4 genes, maybe I think there were 5 genes, right? All of those genes are controlled by this promoter. So if that promoter is not blocked and you can transcribe it, you are going to, remember there'd be the operator right here. Anyway, if you can transcribe one of those genes, you're going to transcribe them all. And that's how you, if you're a bacterium, that's how you get them all together, because there's no need of having one and not having the others. That's not how it works in eukaryotes. In eukaryotes, the different genes here are actually not sitting near each other. They can be on chromosome 1 and chromosome 7 and chromosome 19. They can be spread apart throughout the genome. But you still might need this protein and this protein and this protein all transcribed together. And so how does your cell do that? You have the same enhancers in each of these genes, and they're going to respond to the same transcription factors. And so even though they're spread out, they're not part of an operon. So even though they're spread out in your genome, they're going to respond to the same activators. So these genes can be scattered over different chromosomes, but each has the same combination of control elements. And so they're going to respond to the same combination of transcription factors. Copies of the activators recognize specific control elements and promote simultaneous transcription of genes, even though they're spread apart through your genome. Okay, so next thing we're going to talk about is mechanisms of post-transcriptional regulation. So post-transcription means, okay, we've already made the RNA. What are the other things that we can do? So remember, the whole point of gene regulation is, do we make a lot of a protein, or do we make none of a protein, or do we make a little bit of protein? So we've talked about chromatin structure. We've talked about transcription, whether you have no transcription or high levels or really high levels of transcription. Once you've made the RNA, now we're going to talk about this a little bit. So we've already talked about alternative RNA splicing in the past. You can make this particular mRNA if you cut out this intron and this one and this one and this one, and you actually cut out this. So here's one particular mRNA, but you could splice these exons, right? The exons are the ones expressed. I hope I just said that right. You can use different exons. So if you don't use this one, but you use this one instead, and you put these exons together, then you're going to produce a different protein. So alternative RNA splicing can give you different proteins, even with the same mRNA. So that's another way that you can control which proteins are made. Another way that you can control which proteins are made is mRNA degradation. So that means, does the mRNA last for a few minutes and then it gets degraded right away? Or does it last for a few hours or maybe a few days? And so here is the piece of mRNA that that's going to get translated into a protein. The entire piece of mRNA doesn't get translated, though. There's this untranslated region in the 5' side. This is the side where the ribosome attaches. And then there's the 5' cap with the guanine in it. And then there's this 3' untranslated region, and then the poly A tail. You need the ends here in order for an enzyme to not come over and cut this up right away, so it doesn't look like viral RNA. But this 3' region at the end of the RNA is really important in stability, in the stability of the messenger RNA. If you have these RNA binding proteins, then it helps the messenger RNA to last a lot longer. So the nucleotide sequences that affect the lifespan of the messenger RNA, does the messenger RNA last minutes or days, reside in the untranslated region of the 3' end. Oops, this part right here. I wouldn't necessarily memorize that, but just get the gist that there's a part of messenger RNA that you can use to make the RNA last a lot longer. If this messenger RNA lasts a few minutes, you're not going to make a whole lot of protein from it. If it lasts a few days, you're going to make a whole lot of protein from it. Okay, so we've talked about this, and we've talked about this, and of course, we talked about that stuff earlier. Initiation of translation. So the next idea is that the initiation of translation of selected mRNAs can be blocked by regulatory proteins that bind to sequences or structures of the mRNA. I'm going to do a different video on this later, but the idea is there's this thing called RNA interference. And RNA interference uses smaller RNAs, small nuclear, well, no, it wouldn't be the nuclear ones, small interfering RNAs that can bind to the messenger RNA here and block it from getting translated. So block the ribosome from getting, from attaching to it and doing the translation. I'm going to talk about that in a different video. What I want to do next is talk a little bit about another way that we can regulate which RNAs get translated. So sometimes your nucleus, sometimes your cells will produce a type of messenger RNA, but your nucleus might not know whether you need to translate it or not. So it'll make it, and then there's something else, another regulatory factor that will come back and say, we don't really need any protein right now, or we do. So an example that I have for you is the translation of ferritin. So ferritin is a protein that binds extra iron when there's too much iron around. So what do we need iron for? You might remember that you need iron in your hemoglobin, and that carries oxygen around in your body. So that's actually a really, really big deal, right? If you don't get oxygen to your cells, then you can't do cellular respiration, and then you don't have enough ATP and bad things happen. So the regulation of iron is actually a really big deal. So remember negative feedback. If you have a lot of iron, you really don't need a lot of iron. Actually, high levels of iron are really bad for you. It can damage your pancreas and your liver, and generally it's not a good thing. So you have this protein called ferritin, and the ferritin can bind to the iron and take the iron levels down. Negative feedback. And then maybe you don't have enough iron. In modern days, you can take an iron pill, or we have usually sources of iron. But if you think back to our ancestors, there might have been periods where there was a lot of iron-rich food, and then periods where there wasn't. So if your iron levels go down and you have some ferritin around, the ferritin can release iron and bring it back up so that you can produce more hemoglobin and more red blood cells. Okay, so ferritin is a protein that binds extra iron when there's too much iron around. This is the messenger RNA for ferritin, and there are two sides to it. There's the untranslated region over here. This is in the 5' side. So this is the part that, if this is available, this is the part that the ribosome attaches to, right? And then you're going to make your protein from here. So if you have plenty of iron, maybe let's even think too much iron. If you have too much iron, you're going to want to make ferritin to bring the iron levels down. So in that case, you have this ferritin mRNA floating around. The ribosome attaches to it. This part's called the iron response element. The ribosome attaches to it. When the mRNA gets to here, the AUG, it's going to start translation, and you'll make the protein ferritin. Okay, the ferritin will take down the amount of iron. Okay, let's say, though, that iron is scarce. Let's say you don't have much iron. That would be this case right here. There is something called an iron response protein. The iron response protein will bind to the iron response element. I think I have it defined on the next page. So iron response element is a part of the mRNA that's responsible for signaling whether you're actually going to make the ferritin or not. The iron response protein is a protein that binds to the iron response element. If the protein is on the iron response element in this messenger RNA, then the ribosome can't attach here, and you don't make any of the ferritin. So translation is blocked, and you're not going to have the ferritin around, so you're not going to bind up the iron, which is good because you need iron right now. Okay, coolest thing ever. that's the whole ferritin thing. The coolest thing ever is I have to show you something else. It's called a protein called the transferritin receptor. So let's take a look at that. So here is a cell, and we're looking at the surface of the cell. So here's the surface of the cell. This is the transferritin receptor. So that's a protein that's found on the cell membrane. It would be like this. And it is capable of attaching iron. So let's say there's some iron around, that's going to attach to this transferritin receptor, and look, it comes in by endocytosis, and now you have iron in your cells. So the transferritin receptor takes extra iron in. So let's look at ferritin and transferritin receptor. Let's say that there's low iron in your body right now. If there's low iron, you would want to have less ferritin around, because ferritin will just eat up more iron, and you don't want that. But you would want to have more of this receptor around. You would want to actually increase translation of this transferritin receptor, because that would help you to bring in more iron. So you want opposite effects between these two. And take a look at what this does. With the same thing, an iron response protein and an iron response element, these two mRNAs actually have completely different responses, because the iron response element is in a totally different place. So for ferritin, the iron response element is on the 5' side where the ribosomes attach, and it blocks translation. For the iron response element of the ferritin receptor, transferritin receptor, that's actually over here, not on ferritin though, right? That would be actually over here in the 3' side. And this doesn't affect the ribosome. The ribosome is going to fall off before it actually gets to this part. All it does is increase messenger RNA stability. So it keeps this transferritin receptor around for a longer period of time. So these same two, so the code in the iron response element here, the code for the iron response element is in the 5' side for ferritin, and it's on the 3' side for the transferritin receptor. So this same code with the same protein does completely opposite things for opposite responses for ferritin and transferritin receptor. So that's a case of the DNA makes the RNA at the same level, right? But it's the RNA making the protein that's modified at this point, or that's regulated at this point. Okay, then almost here to the last idea. Once you actually make the protein can be processed. So sometimes once the polypeptide is made, it folds and you have an active protein right away. Sometimes it needs to bond to another polypeptide. Sometimes, though, it's made here in an inactive form. We've seen this with pepsinogen, and then the acid activates it to become pepsin. Here we're seeing it with zymogen. I don't even know if I'm saying that right. But anyway, it's inactive, and there's a piece here that needs to be cut, probably by another enzyme. And then you have these two pieces of the protein that are now close together, and that's the active site for some enzyme. You might also, oops, I don't have it here, but another thing that you can do is phosphorylate an inactive protein for it to become active. Okay, last idea here is protein degradation. Some proteins last for a long time, and some proteins are broken down more quickly. If a protein is going to be broken down, one way to do it is tag it with ubiquitin. So ubiquitin is a molecule that your cells can attach to a protein, and when they're attached, it marks the protein for degradation. So the proteasome is this structure. We looked at that way back when we did cell structure. And if they're connected together, then the proteasome will break down the protein into little fragments, which can then get broken down to amino acids and reused. And that's the story for today.